In this video, we're going to be looking at the process of using a DNA profile, which is one of these images that shows these black bars on it or black stripes. And this is a technique that will identify individuals by analyzing some of the many sections of non-coding DNA. So remember, non-coding DNA is DNA that does not code for a particular trait or characteristic or protein or hormone or anything like that. So they're actually portions of DNA that we have in our body. Most of our DNA actually doesn't code for anything. But these portions of non-coding DNA can be run through a machine, and it's a process called electrophoresis. And what they are looking for is short tandem repeats that we have in common with people that are closely related to us. So you have these portions of your genetic code, which might be adenine, cytosine, adenine, cytosine, adenine, cytosine, and etc., it goes on, of this code of nitrogenous bases that goes on for a while. It repeats, and it's a short piece. And you get those short tandem repeats from your parents. Now, you don't need to know anything about how we get a DNA profile. You need to know that this is a DNA profile. We do not call it DNA fingerprinting. This is an old term, and you will see it in some textbooks, but it is definitely not accepted. If I ask you what this image is showing, it is not a DNA fingerprint. It is a DNA profile. So let's have a look at how we can be asked questions on a DNA profile. So the first question you can be asked is about proving paternity. Now, it's important to know that DNA profiling can also be used to prove maternity, but normally the maternal line is assumed as being correct. So we know as a woman that we gave birth to a child and that was our baby. But yes, baby swapping can happen in a hospital, so it might not be that woman's child, and she might want to undergo DNA profiling to find out. But in this case, the mother is known as being the biological mother of the child. And it's important to remember that you get 50% of your DNA from your mom and 50% of DNA from your dad. So what we look at first is eliminating the bars that the child got from its mother. And for this, you need to use a ruler. So you look across and say, well, that bar must have come from the mother. It lines up perfectly, and it's the same thickness. That bar the child got from his mother, and that bar the child got from its mother. So we talk about comparing the DNA profile of the mother and the child, and you find the bars, the black bars, that the child definitely got from its mother. 50% of the child's bars should come from the mother, and the remaining 50%, or in this case, must come from the father. So now we look at the bars that the child did not get from his mother, and we read across and see which father it could have come from. So we don't just stop at dad too, because we are humans, we do have DNA in common with each other. So all of the child's bars that are not from the mother must match with the biological father. So again, we read across here, and there's a match with that parent, with dad 1 and with that one. And we read across with the last one, and there's a match with dad 2 and dad 3. I say dad in inverted commas. You can't see my inverted commas, my air quotes. So if you look across here, you're finding the man that has all 3 of these bars in common with the child. So this man does not have that one. He has that one, but not that third one. This man has the first one, not the second, but the third. Again, it's not a 100% match there, so he is not the father. Whereas dad 3 has bar 1, 3, 2, and 3 in common with the child. So therefore, he is giving all 3 of those bars in his DNA to his child, and the mother gave the other 3 bars. So therefore, dad 3 is the biological father. So when answering questions like this, you must mention that you are looking at comparing DNA profiles. So you look at the child's DNA profile and identify the black bars that match with the mother's DNA profile. So the word DNA profile must be in your answer, and you must talk about black bars, black stripes, black lines, something like that. Then you find the bars that do not match with the mother, and the child must have gotten those from the biological father. So by using your ruler, you find the man which has bars that match perfectly with the remaining bars of the child, and therefore you have proved he is the biological father. The second use of DNA profiling is being able to identify suspects in a crime. Now, it's important to note, this is just placing someone at the scene of a crime, and therefore they are now a suspect. But that does not necessarily mean they committed the crime. They could have been in that house or the crime scene for another reason earlier on in the day. So it doesn't prove someone as being guilty. It simply places them at that crime scene. So in this example, you've got DNA from a crime scene and you run the DNA profile. But you also have the DNA of the victim. So you need to eliminate the DNA that belongs to the victim at the crime scene. So that bar is from the victim, that bar is from the victim, and that bar is from the victim. Please note that if you use your ruler, you will see that these two do not line up perfectly. They have to line up perfectly. Now we know the DNA on the DNA profile, those bars are from the victim. Now you identify who that DNA belongs to that is left over on the DNA profile. Again, using a ruler, because if you look with a ruler, this man's DNA does not actually line up perfectly with that black bar from the crime scene. This bar is thick, so you need to find a thick bar that matches up with it, and this one is a thinner bar. And as you can see again, this man, suspect 3, it does not line up perfectly. So please use a ruler. Don't just use your eyes and trust your eyes. And as you can see, it is suspect number 2. All 3 of his bars match perfectly with the 3 bars from the crime scene. This is a slightly simpler example of a DNA profile to place someone at a crime scene because there's no need to eliminate a victim's DNA. So here you have your DNA profile from the sample at the crime scene, and these are your 3 suspects that you have. Now, in this case, there needs to be 100% match with someone and the DNA profile. So all of the bars must match perfectly with the DNA from the crime scene. And again, you use your ruler and read across for all of the bars on the profile to see who has matches with this DNA profile. And you read across, and as you can see, it is suspect number 2, once again, who has all of the bars on his DNA profile or her DNA profile, matching all of the bars from the crime scene. So you must get an idea that the DNA profile matches 100%. Please do not say that most of the bars match. If you talk about most of the bars matching, you will not get the marks here because this would be, let's say, my DNA that I left at a crime scene. So it has to be a perfect match to the sample that they take from me.